Yeah,
Okay.
you may not have seen the notice, Edward, but in the state of Maryland, we have to tell people that we are recording them if you record this and get their approval to have their picture on in the recording.
Okay, maybe that can be made at the beginning. That announcement.
Yeah.
Sounds like a good one.
That's the Linda
I'm
trick.
I'm not in Maryland, but
Oh, yeah. That's a pretty interesting issue. You know, what jurisdiction applies? I wonder if DC has a different rule. That's
Well, I was saying when the governor first announced that we were not supposed to travel from state to state, I live in DC, so I was like, I can't go to work anymore.
yeah, right. Now there's all kinds of issues now that are arising because of this. You know, we're still waiting for one committee member to log in, so hopefully that won't be a problem.
I'm gonna go get a cup of tea.
oh, you're in a bunker.
Yeah, I'm very, it's actually a CG bunker.
Oh my God. Johnson. Any response from Yossi?
I just texted him.
On a shirt.
Hmm?
Did you dress up all the way, or did you just put on a shirt?
I am wearing pants. If anyone's worried about that, yes. Yeah.
Good.
Ready, fully dressed up, ready to talk bats.
I mean, it's possible he's not using the right Zoom login. Edward, did you send him? Because I've got this, you know, with the defense.
I'll send him the link again, just to his WhatsApp.
Had this problem before where more than one meeting, people don't log into the right one, and then they're waiting, wondering.
Okay, well, he was last seen on WhatsApp 5 minutes ago, so I assume he's still around.
I bet he's on the other Zoom. Let's hope he appears here shortly.
Oh, the one that you sent out for the
defense
committee yeah.
Well, you know, it's inevitable there will be some little snafu.
I've attended a couple of virtual defenses in plant sciences.
Oh, Yossi just said he's coming. We have confirmation.
Good.
And I was actually amazed at how well it worked.
This might be, you know, the future, right? Because you can reach a lot more people doing this than you could in a normal defense, and people can log in from anywhere. So it's pretty great technology.
Yeah, this is pretty good attendance. thanks everyone for tuning in. And hopefully we'll get started in just a few more minutes.
Edward, I wanted to say I'm not wearing pants in your honor, but I am wearing a dress, so I don't know if that counts.
Thank you. Appreciate the camaraderie.
Right. Anything I can do to support this defense.
I knew a broadcaster, in fact, the anchor man for our local San Jose news, 6 o'clock, who traditionally wore a black suit with white shirt and black tie and shorts. And no one knew the better until they were interviewed by him in the newsroom. But it was pretty funny. It was his tradition.
Hi everybody, I'm connected.
Hey Yossi.
Yay.
How are you doing?
Yeah, we shifted the clock 2 days ago, so it appears in my schedule an hour later. So it's lucky you texted me.
All right. Well, why don't we start? And I'll just begin by, I'm Jerry Wilkinson, Edward's advisor here at the University of Maryland. And I'm happy that this is an opportunity for Edward to give his dissertation defense presentation. I want to just say a few words first about how this will run. We have control of muting everyone. And so I think for the presentation, it'll be best if you all mute. And then at the end of the presentation, you can unmute and ask questions. Or if you have questions as the talk proceeds, feel free to put in a question in the chat box and Edward can try to respond to those at the end. You're welcome to turn your video cameras on. I think that'll give Edward an audience to look at, although he can only see a few people at a time. So certainly do it at the end because I think it'll be nice for him to be able to see everyone who's listened. And, you know, I've been privileged, I think, to have Edward as a student for the last 6+ years. And Edward approached me before he came to school. I was actually in the DC area working at AAAS, wanted to know if there was something he could do with bats. At that time, Cindy Moss was here in the psychology department. And I think because of Cindy, she put Edward in touch with Yossi, who's listening in from Israel. And Edward went to Israel and worked with Yossi's group and began doing a series of studies on bats using technology that Yossi developed for tracking bats and recording their ultrasound in real-time flying animals. It's really revolutionized, I think, the kind of work that can be done. And Edward will tell you all about that. Edward did his undergrad at Princeton, got involved in sort of movement work, actually with Martin Mikulski, working on ants down in Panama. I've only spent a few weeks with Edward in the field, and that was actually on a field course in Panama where we worked on bats and other things. So Edward has done much of his work by himself in extreme conditions, I would say, and has mastered a tremendous amount of technology and computational abilities that has really, I think, created a pretty impressive dissertation from my perspective. So without any further elaboration, I will let Edward take it away.
Thanks, Jerry. Thanks for the nice introduction, and thanks everyone for tuning in. Today I'll be talking about the movement ecology of Mexican fish-eating bats, the species Myotis vivesi. And so movement ecology is kind of a relatively new field of research focused on understanding how animals move in the environment. And so I thought I might start out with an example of some animal movement. So let's proceed. I have to click on the slide. Okay, so here we have an animal movement example. What is Edward doing? Let's look at this track. So it appears that he's first moving south, and then he spends some time sort of slowing down and turning a lot in a certain area. And then he proceeds to go back north and kind of circle back to his original starting point. So we can start to ask some questions about this track, such as, how is he moving? It seems that he's going pretty straight and fast on, some of the main roads, but then when he gets off the road, he starts to slow down and turn a lot. Where exactly are those slowing down points happening? Maybe we should look at some landmarks on this map In particular, we see that there are grocery stores nearby. Maybe that's influential on what's going on. It would be really interesting to know how other individuals are influencing this behavior, especially now that, you know, we're living in this world of really paying attention to other individuals. You know, maybe there's another tagged individual that we could look at the same time and see maybe Edward's like really actively avoiding that person. And are there any other behaviors along the way? You know, we see this park on the return trip. Maybe there's something interesting going on there. And of course, it would be interesting to know sort of, you know, how he looks before and after the trip. And so here's sort of me, the foraging success. No, that is not bags of toilet paper that I have. This is actually me right before I fly out to go into the field. But this is sort of a typical type of track that we might get. And so with a lot of movement, we're interested in understanding how animals explore their environment and successfully find prey. And so here's sort of a typical track from an animal, and this could realistically come from any of these animals shown on the screen so far and be of any of these scales. And so across these taxa and scales, we often see similar patterns and start to ask common questions. And so I'm predominantly interested within movement ecology in understanding how does the resource distribution shape an animal's movement. And so to typically investigate this, we're looking at remote sensing of weather and habitat and how animals are responding to that. And then also, how are animals acting socially? So how do conspecifics, individuals of the same species, other individuals of the same species, influence movement? And typically with GPS tracking, this is done by having many tagged animals and looking at the interactions between those tagged animals. So just to give you an example of some of the GPS tracking technology that's starting to become more prevalent, here we see sort of an example from this paper by Roland Kays showing many different animals tracked with GPS tags, going from the left to the right with cuckoos, tamanduas, sloths, and in the bottom right corner, fruit bats. So there's quite a large range of animals now being actively tracked. And if we look at this map of data from MoveBank, we can see that there's actually been a revolution in the amount of GPS tracking happening all over the planet. And this is mainly because GPS tags have gotten smaller and their battery life has increased, as well as getting cheaper. And so more people are having access to using these tags. And so I just want to kind of describe some of the range of possible studies that can be done with GPS tracking. So here we have collective motion tracking, sort of the hierarchy of pigeon flocks. You could also look at the life history of an organism, looking at maybe the mortality of migrating birds, which ones survive and which ones die and where. We can also look at ecosystem services. How is seed dispersal influenced by these animals? Or the social behavior. This is really interesting work by Meg Crowfoot and now at the Max Planck, looking at group sizes and how that influences who wins in monkey fights. Or conservation behaviors. So much work in animal tracking is looking at conservation of animals and how they might be using corridors, such as with these fishers. Or we can look at ecophysiology and maybe how energy budgets change with regard to their movement, such as how these mountain lions are spending energy attacking prey. But with just GPS alone, often we're sort of missing the context of the behaviors or the social component. And so people have been looking at adding additional sensors to GPS to supplement what else might be going on. And so one common sensor that people add is an accelerometer. So these are very small and lightweight and can give you quite a lot of high resolution information on the movement of the animal. In addition, you can also put video cameras on animals. And this obviously provides a lot of context as to what the animal is directly experiencing. But these are usually restricted to larger organisms. And what I'll be talking about mostly for this talk is adding audio sensors to GPS tracking. And they can provide a lot of the context for how behaviors are happening and really start to make sense more of GPS tracking. So some examples of how audio has been used. Here's some roe deer that had acoustic tags on their mic, had acoustic tags and found that they were more vigilant in environments with higher predator possibilities. So they actually could detect when they stopped chewing. Some of the earliest work on putting audio tags on animals has been in the marine world. So here we have a dolphin carrying a D tag, and these tags can record audio spectrograms of these echolocation calls the dolphins produce. So here we have a spectrogram showing the frequency and the time. And so this is about a 10-second clip, and the amplitude is shown below that. And so first we have the clicks that the dolphin produces. These are individual echolocation calls. And then it goes into this buzz, this foraging event, where it actually tries to capture a prey that's very characteristic in echolocating animals. And more recently, tags have been developed for bats. And so that's what I'm going to be talking about a lot today. So some of these tags have gotten very small and can be placed on echolocating bats. And we can also get the same sort of attack sequence recordings from them. So let's take a step back and have a quick refresher on echolocation or biosonar. So this is when organisms produce sound and then listen for the returning echo to localize objects and prey around them. And so bats are famous for this behavior. And here's a spectrogram showing this behavior. And this first starts out with regular echolocation calls. You see this sort of downward slope call. So this is a frequency modulated call that the bats produce. And then they're listening for the very quiet echo that comes back. And as bats actually focus and try to capture a prey, they often produce what's known as a buzz that I talked about just briefly before, in which the calls become closer together and also drop in amplitude and frequency. And so you can understand why they're called a buzz if I just play the sound right now. So it kind of rattles to a buzz at the end. If we slow that down a bit, you can actually hear each individual call a little more clearly. And so bats are actively telling us their behavior. If we look at sort of this diagram of a bat flying down and trying to catch a prey item from the surface of water, we can see the echolocation calls that correspond with this movement. So in the beginning, we have the approach where there's fairly rapid calls, but still a little bit spaced out, followed by a buzz. And this happens around numbers 4 and 5. And that's actually just before it actually captures the prey. Then you have a silent period where it's actually catching and eating the prey before it resumes to its search calls. And you can see that this all happens in the span of about a second. And so we can actively get examples of foraging attempts from recordings of echolocation. It's also possible to record foraging success, such as when animals are eating very hard-bodied organisms. You can sometimes hear chewing, which is a clear sign of foraging success. It's also possible to record when other conspecifics, again, individuals of the same species, might be present. So here in the tagged dolphin, you can see calls from the tagged animal in red, and then in purple, calls of a nearby calf. And this has also been shown in bats as well, that you can have calls of the focal bat here in dark red, and then circled in red are the sort of lower amplitude calls of a nearby conspecific. And then I also want to point out that you can record other types of calls. So not only are echolocation calls being produced, but you can also have more social calls that might be observed. So here's calls from a dolphin where it's creating its signature whistle calls. And here overlaid on the depth that the dolphin's going since it's been tagged, you can see exactly where it's producing these types of calls. It's also possible to record potential prey. So here are examples of crickets or frogs producing calls in the background. So you could potentially have an idea of what the bats or what the organism might be experiencing from its environment and how that might influence its foraging behavior. But that's not to say that there aren't drawbacks with using audio. Lots of these devices are very energy and memory intensive. Audio files typically are very large, and so they'll take up a lot of energy and memory on the tag, as well as you need to often recapture the tag to get any of the data back. It's usually files that are too large to be transmitted remotely. And with this extra memory and energy requirements, you also have extra weight because you're often using a larger battery or maybe a larger memory card for the tag. And then finally, it's not trivial yet to go through all of these audio files. And so there is quite a substantial time devoted to processing and analyzing these data files. In case I haven't impressed with you enough the advantages of tagging bats, I wanted to first talk about bat diversity, that bats are not this monolith, as you might hear when reading stories, particularly now about coronavirus coming from bats. There's over 1,400 species of bats, and they have a variety of diets and social organizations and complex behaviors with that. And so while most bats are insectivores, we actually have a variety of diets, such as fruit-eating bats, highlighted in green. We have nectar-feeding bats in yellow, vampire bats in red, and what I'll be talking about for a lot of this, fishing bats. So there's a lot of diversity in the bat world, and with that, we hardly know anything about how they're actually exploring their environment. And so my PhD was trying to get sort of more understanding of that by looking at the GPS tracks and movement of bats. And so I focused on the Mexican fish-eating bat, Myotis vivesi. So this is a marine foraging bat, a quite unusual bat. It eats small fish and crustaceans, and it weighs about 30 grams, so large enough to carry the tags that we're working with. And so here's just an example of some of the prey items that have been found around it, small fish and crustaceans. And so our understanding of what the prey behavior is doing is that they're vertically migrating each night. And so during the day, the open ocean is actually quite a dangerous place to be. And so many small fish and shrimp hide in the depths where it's dark, and then at night come up to the surface to feed on algae. And so during this vertical migration to the surface is when the bats are able to intercept. And so fishing bats typically have very long feet, and they use that to scoop prey from the water surface. And so if any of these fish or shrimp happen to be right at the surface, the bats can actually access them. So let's go to our study site. Here we're looking at North America, and we can zoom into a tiny island in the Gulf of California called Isla Partida Norte. And so this tiny hourglass-shaped island in the middle is home to the largest population of fish-eating bats. And so it's a nationally protected island in Mexico, and there's no infrastructure there at all. And for one month each summer, we've been traveling there with this very courageous group of Mexican grad students from UNAM to study the behavior of these bats. And so we bring all of our supplies with us, we take everything back with us when we're done, and we're camping on the only flat part of the island, which is a small beach here. So the bats live under talus slopes, which are these loose piles of rocks. And so here I am searching for bats. And I just like to take a second to acknowledge my main collaborators. So Gerardo Herrera from UNAM has been studying these bats for decades. Working on this island has been essential for all of our work. And Yossi Ovel, as Jerry mentioned earlier, developed the tags that we've been working with. And so just to give you a little bit more information about these tags, these are called Vesper tags, and they're multi-sensor tags. They actually have capabilities for more than just GPS and audio. But what I'll be talking about today is just GPS and audio recordings. And here we can see some of these GPS recordings and very high resolution tracking of a nectar bat on the US-Mexico border. And as well, they're recording audio from these bats as well to look at where exactly they were foraging. So for my project, we're focused predominantly on tagging lactating females. And so the importance of tagging females with pups is that it ensures recapture success. So as I said before, the tags don't transmit data. We actually have to recapture the animal with the tag or find the tag on the ground to get any data back from it. And so here, what might look like a big growth on the side of this bat is actually the pup tucked under the wing. You can see its feet sticking out here. So this is how they'll often nurse the pups. And when we find them, the mother and pups are together. And so lactation is a very demanding part of the demanding period for a female. And so one other interesting point is that foraging flights that we record should be efficient. We wouldn't really expect too many other behaviors when a bat goes out. It's really just trying to go out and find as much food as possible and come back and try to raise this pup. And we've also looked at how tagging bats might influence the behavior, the health of these bats. And we found little effective GPS tagging on mothers or pups when compared with a much smaller just radio telemetry tag. It doesn't really seem to affect the weight of either mother or pup or the amount of time spent foraging. So here we have a bat with a tag that's been released into the rocks. So they're attached using surgical glue to their backs. And then the microphone, in this case is an older version, the microphone comes up over the shoulders. So we release the bats, and then the next day we return and try to triangulate using radio telemetry to find exactly where the bats went. So in general, this is what a lot of our data looks like. We have tracks colored for each individual, leaving Isla Partida in the north and often going to this southwest region. We often tag bats between 1 to 4 nights per individual. And we're trying to get fairly high frequency GPS sampling so recording typically every 15 seconds. And we have GPS and audio data collected over the past 5 summers. So getting into sort of my research questions for my PhD, the first question was obviously, what exactly is the animal doing? You know, how can we make sense of these GPS tracks and kind of parse the behaviors? And to do that, we use acoustic validation of behavioral segmentation methods. So there's different methods to chop up the tracks, and we try to validate exactly what's going on. Then we ask, you know, what's unique about where we find foraging sites? We ask, where is the animal going, and try to do some habitat selection of these foraging sites. And then we ask, when are the bats social? Why would you want to be social? And to do this, we actually do a multi-species comparison. So we look at 5 different bat species and try to ask questions about what predicts social foraging. And then we also ask, are there any other behaviors maybe we could potentially explain recorded with these tags? And indeed, I found an in-flight social call, and we try to test different hypotheses for what the function of that could be. So our first step is, what is the bat doing? Can we interpret the movement phases of this track? So here we see a foraging flight of a fishing bat going out. It goes generally fairly straight and fast, turning a bit. And then sort of at the bottom of the track, it spends a lot of time slowing down and turning before returning back up. So taking a step back, GPS tracking data is just localizations in time and space. But when you zoom out, you often try to see these phases or behavioral bouts that might be happening in the movement. And so we might define these as, you know, searching for food or perhaps escaping a predator. But how do we really know what's going on with a given track? So GPS segmentation typically assumes that foraging is what's known as area restricted search, where an animal slows down and begins turning a lot more. And so if we're trying to define foraging on a track like this, we would probably highlight these areas circled in red as foraging, where the animal's behaviors change. It's turning quite a lot and likely going slower. And then commuting is sort of a more general behavior that we could probably describe as often when an animal's going fast and not turning very much. And so we would say this area in between these 2 foraging events is likely just commuting between them. Another question we have is, you know, can we really connect this audio data that we're recording with bats with GPS data? So audio data that I showed before is, you know, happening in the time span of milliseconds, whereas the GPS tracks are happening over several minutes to hours. And so when we're thinking about bat behavior, you know, can we make that leap? Do we see bouts of behavior lasting sufficiently long durations? So let's return to the track that we had before, and let's overlay some of these feeding buzzes that we've found in the audio on the track. And so we see that some buzzes occur on the outgoing track, but the majority of them happen during this area restricted search, when the bat begins to slow down and turn quite a lot. And so that gives us some indication that they're likely using an area restricted search for foraging, and that segmentation methods should apply fairly well. So I'm not going to go into the details of exactly how segmentation methods work, but I'm going to give you some examples of how they might be different. So here we have a GPS track where we have different GPS fixes over time going from the start in the left to the end in the right. And if we overlay sort of a segmentation method, it might say that the foraging behavior is just these 6 red points in the center, a fairly short segment, and then the rest of it is just commuting. Whereas another method might be a little more generous and say, oh, actually the first 2 points before that are part of the same segment. And additionally, this point toward the end where it seems to slow down for just a moment is also another foraging bout. And so how do we know which method is right for our data? So we can try to validate the methodologies. And actually few studies really do take this extra step, mainly because it's hard to do. So in some cases, you can directly observe the animal's behavior, such as these researchers looked at kill sites and would actually track the wolves and find exactly where kill sites happen to better parameterize their methods. Of course, you can use other sensors like onboard video. Here we have 3 gannets being tracked with GPS and video. And the videos show different behaviors, such as diving with fish, flying near other birds. And in the bottom right, you can actually see that this bird actually spent a lot of time following another ship, which would have been very difficult to parse out just from GPS tracks alone. Other methods use time depth recorders, so it actually records how long and how deep an animal dives. And so from another study on gannets, they actually tried to calibrate how well different segmentation methods worked on predicting these diving events. So I'm attempting to do something very similar. We're going to use the buzzes to validate different segmentation methods. And so we use a subset of individuals where we have good GPS and ultrasonic recordings. And so we just have 11 individuals for that. Here are the tracks of those individuals with the buzzes overlaid. And in general, buzzes occur at lower speeds and higher turn angles than most of the track. We only found about 648 buzzes out of almost 40,000 audio files. So buzzes are also kind of rare. Okay, so moving on, here are the segmentation methods that we used. In general, they fit into 3 main categories: pattern description, process identification, and change point detection. The main point I want you guys to realize is that as we go down the list, the methods become more complex and there's more parameters that you need to tune to get them to work properly with your data. So the top ones are kind of the easiest ones to use, the bottom ones are more difficult. And so importantly, these results are not all doing the same thing. When we apply them to our data, they give very different estimates for how much of the track is actually foraging. The first 2 methods in dark green suggest that not that much of the track is foraging, whereas the other 3 methods are fairly consistent. Around 50% of the track is spent foraging. And then if we look at how long is a given bout or segment length, the first 2 methods say that an event is only happening for maybe a minute or 2, whereas the other ones have it around 5 to 7 minutes, and the last one has it almost half an hour. So there's quite some differences in how these methods are being applied. So let's look at an example of how they actually map onto a track. So here we have a track on the right showing, again, the same track with the feeding buzzes occurring, and then the majority of the foraging events happening down the bottom. Here we've overlaid each segmentation method's definition of what is foraging. And so in general, they all agree that the middle portion of this time is showing that a foraging event is happening. Sorry, I forgot to explain these 2 graphs on the right. This is showing turn angle. So this is bounded between 1 and -1. Essentially, when it's at 1, that means that it's turning very little. And when it deviates from that, that means it's turning quite a lot. And so here we have commuting where it's not turning very much, and then it begins this area restricted search where it turns quite a bit before commuting back home. And below that, we have speed showing fairly high flight speeds during the commute, and then it slows down considerably during this area restricted search before speeding up again for its return. And so the methods generally agree, but we do see some differences exactly how they parse that foraging event. Some are very generous and say the whole track should generally be defined as foraging, whereas others are a bit more conservative and just say very small portions should be. And so looking at overall performance, we can see that hidden Markov models perform best. So here we have a plot of the balanced accuracy, which is an average of the true positive rate and the true negative rate. So how many buzzes actually occurred in foraging labeled locations and how few buzzes occurred in commutes. And so we're trying to maximize that. And we can see that hidden Markov models have the highest average balanced accuracy measurement. So that's the best method. And although they're not significantly different from first passage time or CVCP, we can see they are significantly different from KMC and EMBC. And overall, buzzes are rare, as I said before. So that's why these performances aren't necessarily that great. They're slightly better than random, which is this dotted line at the bottom. But overall, we can rely on hidden Markov models consistently doing a good job. So can we accurately say what the bat is doing? Yes. We can say that these bats are using area restricted search, that hidden Markov models are the best performing methods. Now we see these behaviors happening for fairly long sustained periods. And methods that seem to do well suggest that foraging bouts are happening for a few minutes, approximately 6 to 10 minutes. So now that we have a method to say where foraging is happening, let's ask what exactly is going on in that environment. So how do animals select their environment? We typically talk about selection as use disproportionate to availability. So if everything's the same, you can't really tell the difference between what an animal is choosing or not. But if there's some heterogeneity to the environment, maybe an animal selecting some type of resource more than another type. Another main question understanding how animals are selecting the environment is kind of parsing what is used versus what is available. And so we can have different levels of selection, sort of depending on what scale we're looking at. Here, I'm going to be talking about third order selection that is looking at an individual's home range compared with its actual used locations. We also looked at more fine scale selection, comparing where it's foraging versus where it was commuting. But spoiler, the analysis doesn't really come out as well. The predictive ability is quite low for those models. So what factors might predict marine predator movements? In general, the predators are searching for prey, but the prey are likely influenced mainly by the environment that they're in. And so while we don't have a good ability to track the prey, we can try to use the environment to understand how the bats might be exploring their environment. So just to give you guys a little bit of information about this environment, ocean depth is a really important aspect to this area. So the island, again, is pointed out by this yellow arrow here, and next to it is this very deep channel called the Ballena Salsipuedes channel. And so this area is known for having very strong tides. So locally, it's known as the Strait of Hell, because these very strong currents get squeezed between all these islands and then also mix when it comes to this deep channel. And so this mixing in the water column creates sort of this continual upwelling activity going on in this area, increasing general productivity. And so if you look at monthly average maps of sea surface temperature and chlorophyll, we see that this area is generally much colder than the surrounding area and has much higher chlorophyll abundance. So it's locally known as a very productive area for fish. So for our analysis, we looked at 2 static covariates, such as ocean depth or bathymetry, slope, or distance to land explain where the bats might be going, or maybe do more dynamic covariates explain where the bats might be going. So we also looked at this chlorophyll and sea surface temperature. We used 30-day averages, mainly because a lot of data was missing due to cloud cover for finer resolution. So in general, when we're looking at the tracks, we see data from 2015, 2016, and 2019. And in 2015 and 2016, the bats typically depart to the southwest. So we have 19 individuals departing to the southwest here. And Isla Partida Norte is positioned here, just for reference. We have 29 individuals flying in 2016, and some went off to the north and a few over to the east, but in general, we see the same trend. And in 2019, the bats were less restricted in their departure directions and seemed to go in all different directions. We did a test for whether or not they were randomly departing the island or not. And in 2015 and 2016, we found that they definitely have some directionality to where they're going, suggesting that there's some selection in how they choose where to forage. So our models to look at habitat selection, we started with an individual track, and we took the foraging locations and extracted those, and then generated random locations going in any possible direction from the island, and any distance between the closest foraging bout and the farthest foraging bout of a bat. And so this gave us sort of our null set to compare what was potentially available to a bat versus what they actually used. And so we combined models of static and dynamic, sorry, that came out of order. So the static parameter models did better than dynamic parameter models, but in general, combined did the best. So we tested whether static parameters, just ocean depth and seafloor slope and distance to land were influential, or just chlorophyll and sea surface temperature were influential. But actually putting all of the parameters together seemed to produce the best model for explaining where bats were going. That came out better. And so if we look at some of the selection that the bats were using, here it shows seafloor slope. So they were showing this polynomial relationship with where they went to forage. They weren't selecting the steepest slopes, they're actually selecting somewhere in between. And for ocean depth, they tended to choose sort of the deepest areas as well. For chlorophyll, we saw that there was also there was a polynomial relationship where they weren't choosing the most extremes, they were choosing somewhere in between. Often it's hard maybe to find the most extreme of different resources. And for sea surface temperature, they're generally choosing some of the lower sea surface temperatures. So when we look at where they're foraging, here first are maps of the observed foraging locations rasterized. And so we have an idea of foraging site count per year, just to give the GPS tracks another view. In general, we can then compare these with where we would predict a bat to be foraging on a given day for each year. So when we look across years, the bats are foraging predominantly in this Myotis channel to the west of the island. And while we see some differences between years, in general, they're fairly consistent. And so what we presume from the habitat selection models is that these foraging sites are likely consistent with tidal upwellings. But choosing this area with a very deep channel is important because the currents are likely causing these upwellings continually in this area, creating a lot of productivity for the bats to benefit from. But I just want to point out that we might not have the full story. So when we did collect data, they occurred during these periods where these black dotted lines are overlaid. And these all correspond with an El Nino event. So this is a multivariate index of the El Nino cycle, El Nino Southern Oscillation. And when the graph is sort of in the red section, that means that it's an El Nino year. And when it's in the blue, that means it's a La Nina year. And so in 2015 and 2016, we collected data during a very strong El Nino event. And then in 2019, the data was during a fairly weak El Nino event. And so El Nino has a very strong impact on the Gulf. During El Ninos, in general, the whole Gulf is a lot warmer and has a much reduced productivity. And so this can be detrimental to seabirds who can't get access to as many fish, and many marine mammals also avoid coming into this area. Whereas during La Nina events, we have often sardine schools exploding in abundance and a much healthier ecosystem. But during this period, what we often find is that the sea surface temperature in this Myotis Celcipotis channel remains consistently much cooler than everywhere else. So it could be a refuge that a lot of animals go to during this time period that they're able to still survive. So because this island is right next to this very productive area that's consistently good, that might explain why the bats are consistently going to this area. And it'd be interesting to look at what exactly is happening on other years that might correspond with the La Nina. And so just to summarize foraging site analysis, we found that foraging directions are not random. They don't always leave the island in any random direction, especially in 2015 and 2016, they tend to go to the southwest. That when we looked at whether static or dynamic models were better, we actually found that combined static and dynamic models do the best, and that bats are choosing areas that seem to be in this Myotis channel. And that between years, at least for the 3 years that we have data, the patterns seem to be consistent. Okay, so moving on to the third section, when should you be social? So bats are known to be very gregarious in roosts, with some roosts having millions of individuals. So what exactly happens when the bats go out to forage? Do those same bats that are very gregarious roosting also show the same patterns when they're out foraging? So we can try to look at some of this behavior, but first let's talk about what we might expect. So individuals could be completely solitary, where they travel alone and they're solitary foragers. Alternatively, bats often share a roost, so they could use information centers, whereas they're paying attention to individuals coming back that are full, and they can then use that information to learn where exactly to go to get food. Or we might expect local enhancement, where they're foraging, and if they encounter another close individual foraging, they can benefit from that and also try to cheat off of that individual. Or we might expect that the individuals are traveling, searching together as a group, and cooperating to benefit from finding prey that individually they would not be able to find on their own. And so there's been considerable interest in understanding sociality in bats, mainly because echolocation broadcasts a bat's behavior to nearby individuals. So the same that we're able to record a bat's behavior and understand what's going on from echolocation calls, bats can do the same. And even there's further evidence that group foraging in some bat species might be prevalent. One prominent example is from my advisor, Jerry Wilkinson, who found that social calls can actually coordinate group foraging in greater spear-nosed bats. So they produce this screech call that attracts other individuals to come forage with them. And then finally, radio tracking from Dina Deckman reveals that some bat species forage close to roost mates, which might be evidence of this also of group foraging. But there hasn't really been direct evidence showing the tracks of the animals of bats flying close together and foraging so far. And so just to break down exactly how eavesdropping might occur in bats, in general, bats are producing echolocation that's this very, very loud call. But the range for echolocation is actually a fairly small distance because they need to produce this really loud call and then listen for a very quiet echo coming back. And so the range for an echolocation can be fairly small, tens of meters or about 10 meters, whereas they might have a slightly larger volume where they can detect larger obstacles, something that might reflect a larger echo or more amount of the echo. But for a nearby bat flying by, they can actually hear the bat calling from quite a large distance. So up to 160 meters away, you can hear another bat calling. And so what might be going on is that as bats are flying around searching for insect patches, if they remain within this eavesdropping distance, they can all benefit when one happens to find a patch of insects. And so this is kind of referred to as the potato chip effect, that imagine being in a dark room and someone opens a bag of potato chips. Everyone else in that room immediately knows that they just found food and are eating the food. So you don't necessarily know who found the food, but you know that there is food available. And so in 2013, working with UC of L before I started grad school, we actually looked at social foraging in a bat, Rhinopoma microphyllum in northern Israel. And we found, using UOC's tags, that foraging events actually seemed to increase in frequency after bats were near conspecifics, suggesting that they were benefiting from being around other bats. And that seemed to help them find these patches of food. So if we look at my data, we can see some tracks of foraging bats with buzzes and conspecifics overlaid. And so the buzzes are in yellow and conspecifics are in white. And what I want you to notice is that some of these conspecific calls are happening during commute. And so this suggests that the bats are encountering quite a lot of other individuals while they're out foraging. And one other important point is that the bats can likely hear each other much further than what the microphones can report. So the range of the microphones on the bats is likely about 12 meters, whereas the range of the bat's hearing could be over 100 meters or 160 from that previous figure. So they're likely attending to other individuals flying nearby and paying attention to that behavior. And if we look at the conspecific presence overlaid on all of the data we have, we see that it's pretty clear that bats are often not alone when they're foraging. And so this is exactly how we determine that, by looking at these spectrograms of the main bat echolocating. So here in white, we have arrows pointing to the calls from the main bat that are much larger, or much deeper red and have harmonics. And then nearby bats, or conspecifics calling, are highlighted with these orange arrows. So we can see exactly when another bat is around conspecifics. Then we decided to do an experiment to look at whether the bats are actually attracted to conspecifics. So we did playback experiments where we did 3 different treatments showing a white noise control. So just of the same frequency spectrum as an echolocation call, similar repetition rate to see if there's if they're just attracted to noise at all. Then we did search calls, and then finally a feeding buzz. And in comparison to the attraction to a white noise control, we found that search calls were 5 times more attractive, and that feeding buzzes were 15 times more attractive. So they're generally attracted to each other's search calls, and they're very attracted to each other's feeding buzzes. So with my collaborators, we decided to look at 5 different species of bats. So the first 2 are the ones that I talked about just now. So the fish-eating bats, Myotis vivesi, and these are Rhinopoma microphyllum that we studied in northern Israel. Yossi's lab is also working on Myotis in Bulgaria, and these bats are foraging for terrestrial arthropods, so large beetles that are in the grass. There's also nectar feeding bats, Leptonicteris urubuenae. They're feeding on cactus flowers. And then Rosetus aegypticus is a common bat in Israel, and they feed on fruit trees. And so we wanted to look at this hypothesis that differences in prey predictability and distribution led to variation in foraging strategy. So particularly looking at movement and sociality. And so in particular, we're interested in how the predictability of the prey might influence where exactly the bats are, or how the bats are moving. And so just to give you guys a view of some of the tracks, here we have the top two tracks showing these circular meandering paths from the two individuals. And then now on the bottom, we see much different tracks that are often very straight, long commutes with a foraging event sort of at the end. And so in general, these bats on the bottom have very predictable prey. While these Myotis bats are searching for terrestrial arthropods, in general, it's been shown that they're fairly predictable in abundance for a given site in Bulgaria. So it's, you know, it's a predictable resource, whereas searching for swarms of insects or schools of fish is very unpredictable. And then of course, looking for a tree is very predictable. If you're just searching for a cactus or a fruit tree, you know exactly where it's going. It's not going to move. And so when we look at the onboard audio, we find that conspecifics are frequently present in the commuting flights of these bats, and that we did not find any conspecifics present in the foraging flights of these bats. And so again, just reinforcing the point that the top bats have unpredictable but shareable food sources. So they're often finding very large aggregations of prey. If you're finding a school of fish, it's likely more than one single bat could eat, or a swarm of insects, it's likely more than one single bat could eat. But for these predictable or non-shareable food sources, we don't find any relationship with searching and conspecific presence. So the results demonstrate that ephemeral foragers likely use eavesdropping to find prey, and they're likely moving in groups to go out searching for food. Okay, so the last bit, we're going to be talking about the function of social calls. So I happened to stumble across these social calls while going through thousands and thousands of audio files, and we wanted to see if we could figure out what exactly was going on with them. So let me first just play it for you. Here we first have an echolocation call, followed by the social call. So it's this U-shaped call, followed by another call that looks very similar to the social call. When we scrutinized all of the audio from tagged bats, we found that 17 of 31 individuals who recorded audio had social calls present. So it seems to be a fairly common thing with the bats that were tagged. So we wanted to try to understand why exactly they're producing these calls. So in general, there's actually been some pretty phenomenal research showing that echolocation calls can already convey quite a lot of information. So researchers predominantly use echolocation calls to identify the species of bats, but experiments have shown that bats can recognize the sex of a bat from its echolocation calls, as well as the age, reproductive condition, and identity. And so there's quite a lot of information in an echolocation call, and it makes sense if you sort of think about someone's voice. You can often recognize quite a bit of the same information just from hearing someone's voice. So social calls, contrarily, should communicate something else. So they might communicate intent, like I'm going to do something, maybe this is a warning, such as stay away from my food, or solicitation as saying join me to find some food. Alternatively, social calls could convey quality. One example of this could be bird songs, and several bats actually produce songs as well. So they're trying to create something difficult to produce and maybe an honest signal of their quality. Or you could maybe be communicating some additional information about your identity that might be lost over large distances. So maybe it's a more robust representation of who you are, kind of like a signature whistle or something. So here we have 3 different possible functions of social calls. So we have conflict resolution, group formation, and parent-offspring communication. I omitted mating calls from this because we're working with lactating females, and so they likely don't have any interest in mating during this time. So we're just going to focus on maybe the calls are functioning for one of these 3 purposes. So we can try to look at the context and the impact and the information of these calls and what we would predict given each call type function. So if we look at the flight context, we expect that conflict resolution calls would happen near feeding. So you're only really getting into a fight with another bat if you're competing over food. Group formation calls, if you're trying to get other individuals from your group together, that might happen near feeding, or it might happen close to the roost. Whether you're leaving or returning to the roost, some bats like to sleep in the same vicinity of each other, so that might be when they're coming together, or when they're going out to forage, they might try to aggregate to forage together. And then one type of call that's produced seemingly by all bats is this parent-offspring communication. So recognizing your pup is really important, and we would expect it predominantly to occur when they're returning to the roost. We could also think about the seasonal context. So for the first two types, we would basically expect them to happen all the time. You know, you're always feeding or you're always searching with a group, so you would always produce these types of calls. Whereas parent-offspring communication, we'd expect only to happen during early pup volancy. So when the pups are starting to leave the roost, then you might expect these calls to occur. And what impact might these calls have on other bats in the vicinity? We expect conflict resolution calls to actually scare away bats, so decrease bat activity, whereas group formation and parent-offspring communication would likely increase the abundance of bats because they're trying to solicit some response from another bat. And then the information content. What exactly are these calls trying to communicate? For conflict resolution, we would assume that there's some relationship between sort of how strong and big the bat is and its calls, whereas other calls might be reflecting identity, so who you are or who your group is. And so we might expect identity from group formation or parent-offspring communication. So getting into how we exactly analyze this, one method we used was actually to record bats flying in the wild using ultrasonic microphones. So this is kind of a standard way of recording bat behavior, except we took it to another level where we actually put the microphones out in the ocean. We put them on these buoys. And so at the top of this stick is a microphone, and below it is the actual box where the recorder is. And then this is all floating. This is amazing technology developed by our Mexican fishermen that really took care of us in the field, and Dave Johnson, who had the equipment. And so with these, we could look at, in general, when are bats producing social calls? So here is just a bar graph showing the number of echolocation calls that we find in dark gray, and the number of social calls that we also find per hour, per day in pink or purple. And so one of the main findings that we found was that these social calls are occurring later in the night. That if you look across nights, predominantly these social calls are happening towards the end of the night. If we look within GPS tracked bats, here we have 4 different tracks of bats with social calls produced, and the top 2 show the social calls occurring right as the bat returns to the roost. So the track goes from green to blue, so green being the start of the track and blue being the end. And so in the blue portion is where we see these social calls occur. For the bottom 2, we actually see some social calls occurring quite a distance from the roost. So these are all in-flight social call events. This call in particular happens about 6 kilometers away from the roost. But consistently these calls happen as the bats are returning back to the island, so in the blue portion of the track. We can further look at the context by looking at the time between a social call event and the nearest buzz, or the nearest time that the bat's in the roost. And it appears that the social calls don't occur close to buzzes. They're actually much closer to roost events. So the average distance, the average time between a social call and being in the roost is a few minutes, whereas it's almost an hour between when a social call occurs and a buzz occurs on average. We could also divide the track into the percentiles of the track. So 0 being the beginning of the track and 100% being the end. And the majority of social calls, again, happen towards the end of a track. One other interesting finding we found is that the probability of finding a call in a given track increases with pup age. So we are able to calculate pup age from estimates from the weight and the forearm length of pups. And what's handy is that bat pups tend to grow linearly in both weight and forearm length for a certain period of time. And so we use that to actually back calculate how old the pups likely were. And we found that as pups were older, you were more likely to find these social calls occurring. We also looked at the discriminability of these calls using a discriminant function analysis. And so we used just a subset of bats where we had at least 8 calls. So we had 8 individuals with at least 8 calls. And we could discriminate individuals. We had about 85% accuracy for all calls to individual using this 50-50 split between training set and test set. So there was individuality to the calls, although if you look at this plot of sort of how the calls cluster in space, there is some overlap. So you can understand why it wasn't perfectly accurate. We also did playback experiments similar to the previous section. We playbacked buzz calls as well as social calls, and then we used silence as a control. And there was no significant difference between silence and social calls. So we consistently had buzzes being very attractive, but social calls were not much more attractive than silence. So we didn't really find any response to playbacks of social calls. So if we go back to our original predictions for each type of call and look at our results, we see that the context typically is when bats are returning to their roost. The seasonal context is that bat calls increase with pup age. We find no social impact on these calls, and the calls are likely conferring identity. So our best guess for what exactly these calls are functioning for is that they're parent-offspring communication. But these recordings and all of this data was kind of collected opportunistically. We weren't setting out to study social calls. It was sort of analyzed after the fact. And so more work is needed to actually look at interactions between mothers and pups where we can directly observe who is who and know that these calls are influential when mothers and pups come back together. So we're still working on understanding exactly what might be going on, but it's very suggestive that they are parent-offspring communication calls. So just to conclude, the first section we were looking at foraging segmentation. We found that bats are using area-restricted searches when foraging, and that hidden Markov models have the best accuracy. We then used that to define foraging sites, and we found that foraging sites are mostly occurring in this Byenes channel, and it suggests the bats are selecting environments that are consistent with these tidal upwellings, that's where the prey tend to be. And for looking at social foraging across multiple species, it's very suggestive that resource distribution best predicts when a bat is socially foraging, and that patchy, unpredictable prey can create these conditions for social foraging. And then finally, we looked at social calls, and these are mainly occurring on the returning flights. And it's very suggestive that these calls are directive calls used to communicate with pups. So these mother-pup interaction calls. So of course, there's many more questions to ask about these bats. I would love to have lots of grant money and more time to keep working on them, and particularly to look at how do the bats actually respond to their environment during different seasons or in different locations. We were really fixed on just studying the bats during one season in one place, but even looking at different El Nino cycles for the same area could be very informative of how the behaviors might change. What happens when you're at a location with these bats that has a much lower abundance? So this is the largest population of these bats, but some islands have a much smaller density of bats, and that might be very consequential when you think about social foraging. If you can't easily eavesdrop on your neighbors, how exactly do you go about finding food? One thing I never addressed during this talk is actually how the bats detect prey. We tried many experiments to really look at this, but never came to a complete determination of how they really detect prey when they go out searching. Obviously, they're relying on other individuals to help find food, but one bat in the beginning has to find the original prey. So there's many different theories about how they could actually be selecting the prey, and there still needs to be more work to really definitively say what's going on. And for the social calls, while they seem to be occurring in this mother-pup interaction, a lot of the calls were happening before pups could actually fly. And so why would you expect to have these calls happen so many kilometers away from the roost? It's possible that the calls have more than one function, and it would really be interesting to figure out exactly what's going on with all of these calls. Okay, with that, I would like to thank my many collaborators. I would thank the Wilkinson Lab, Jerry Wilkinson, been a tremendous advisor. Danielle, Jerry Carter, and Kim have been excellent, keeping me sane in the lab. The Fagan Lab has also been an invaluable resource. I would particularly like to thank Bill and Ellie for all of their help through the years. My collaborators at Tel Aviv University, Yossi, this project would not be possible without him, and Stefan for all of his help in the field. It was really helpful. And Unam, Gerardo, Andrew, Ricardo, Jose Juan, all of you were extremely influential. And then I had a whole army of undergraduates that helped me along the way, processing audio, really going through all this data, and incredible funding sources. I applied for every single grant you could think of. and many of them gave me money to ask crazy questions about ridiculous bats, and I thank all of them for their funding. With that, I'd be happy to answer any questions. Whew.
Yeah.
Thank you.
So if everyone would be, if you want to say anything, unmute and feel free to ask a question. You can raise your hand if you have the participant box open. Maybe you should close your screen, or
Okay, yeah, I can stop sharing.
then you can actually just see everyone.
You can
Yes.
raise your hand also in the chat window, it's in the participants list, and then someone can coordinate who.
Yeah.
Oh wow, there's a lot of people.
So
Hey everyone, I did not look at the participants for the past hour, so thanks for coming.
So there were a couple chats already, questions that were asked. Kevin actually asked, do you have any anecdotal info from local people about good fishing areas? And does that have any relationship to where the bats are going?
So yes, the fishermen that we worked with to get to this island would basically spend all of their free time fishing in this area. It's not that easy to access. It's about a 3-hour boat ride from the nearest town. But if you can get there, it's usually very worth it. And then we happened to be there during the La Nina year, but we weren't able to record GPS tracks. But that corresponded with this really huge boom in the amount of sardines. And so we saw a lot of sardine fishermen coming into this area. Even though it's actually a protected area, there was a lot of interest in foraging in this particular area.
Cool, thanks.
Kristen has a question, but maybe she could ask it directly. And there's actually...
Sure. Can you hear me?
Mm-hmm.
Yeah.
Yeah, and now I can't remember what my first question was. Oh, yeah. So it wasn't clear if you, are you able to determine how, if these calls are cumulative in their effect potentially, like the frequency of multiple individuals changes the overall sound, and if that might be something that is attracting certain individuals, if they're all hearing a louder call from a certain region, if that's something that's attracting them because it might be an indicator that there are more foragers at that site, for example.
Yeah, I think that's interesting that if you have multiple callers or multiple echolocation calls, sort of hearing a crowd versus just hearing one individual, how does that influence your behavior? We didn't particularly look at the effect of density on playbacks, but I think that would definitely have some influence on the results. That if we recorded just a ton of bats calling, that would likely have a similar effect or more attractive effect. Kind of like when you see a lot of people looking a certain direction, you're more likely to look that way as well, this kind of collective behavior. And you could also imagine that just more bats calling is also louder. And so you're more likely to detect it from a further distance. So we haven't directly looked at that, but I would suspect that that's also influenced.
Batty Chica.
Sorry, that's my computer deciding that. I have several questions, but I'll start with one that's related. I'm not really sure you can distinguish between active social foraging and local enhancement effects. So what do you think you can?
So it's definitely difficult to tell whether animals are searching in a group or just sort of stumbling upon the same resource, or maybe eavesdropping when they happen to be close and copying. And so what my collaborators did was actually simulate what might be happening if you have sort of no general attraction, but they all end up at the same foraging events by chance, or they do show some attraction. And in general, we wouldn't expect bats to encounter each other as much during commutes as we find in our audio. So they're running into each other more than you would expect by random chance if they're just kind of moving at random. And so that's evidence that they are attracted to each other. And then the playback experiments also suggest that just search calls show some attraction. So, it's hard to really describe what a group foraging would look like. It's probably looser than a flock of birds, but they seem to be paying attention to each other and moving together in a way that you could define as a group foraging.
So
Should I
that's
ask
the
more questions?
Yeah, go ahead.
Sure. Well, first, a comment maybe. There's no just about local enhancement, right? I mean, if they do restricted area search, then they also have an increased chance of encountering each other if that's like the high production area. Then, I mean, if they all go to an area that they know is rich and then listen to each other. But yeah, so my next question was how you calculated those. Is that based on the rhinophila? Because I cannot imagine that a Myotis call carries that far.
So you were asking, sorry, I cut out a little bit, but you were asking how we calculated the distance that the echolocation calls could travel. I didn't personally actually calculate that. Yossi's lab did, but in general, and we also did not calculate exactly how loud the bats were, but compared with other bats we use the same tags for, these bats were typically much louder, like they clipped the microphones, meaning that it exceeded the volume threshold that the microphones were really adapted for. And the calls are also happening over water, which is sort of this acoustic mirror, and it is more likely to almost double the distance that sound can travel because it reflects off the water surface. So it's likely that the bats can hear each other from quite a large range, but we do not have precise calculations on how far.
That's pretty cool though. I mean, that's very far.
I see Eileen and Rui have a question. I see a hand up over there.
Yeah, you're on.
Yeah, my question pertains to the one of experience and memory in these bats. So you said that both static and dynamic models, combining them works best to figure out where the bats will go to forage. You could imagine that the static thing could be a consequence of memory and experience. Good foraging spots are usually the same place over and over and determined by static characteristics, but then you have to adjust dynamically. So is that what you believe? And can you age your bats? Can you tell which ones are more experienced? And does that have any effect on their foraging routes? If they like a good old fisherman versus a novice one, how do they take account of all this information?
Great question. So we weren't able to track the bats over long periods of time. We really just had a few days. So we weren't able to really look at what their previous experience was. But work by Yossi Ovel right now on tracking pups flying out and doing their first flights for several weeks really is starting to look at how bats learn and figure out where to go. But in general, we didn't really have much information about their experience. So yes, it's likely that static environment is easier to memorize. What we did look at that I didn't quite talk about is that foraging locations for the same individual between night typically are not that close together. So for some of the predictable foragers, they're maybe 10 to 50 meters away from where they foraged the previous night, whereas the unpredictable foragers are often about 4 kilometers away from where they were the previous night. So they're not necessarily just returning to the exact same spot, but it kind of also comes back to what scale. So, from a large scale, they're going typically in maybe the same direction night after night, but they're not always stopping in the same place. And it makes sense that the ocean is also just this unpredictable environment. Depending on the tides, you might have upwellings occurring in different locations. And so that could really influence where exactly they're going. Getting to the aging question, that's actually something that my current advisor, Jerry, is working on, using DNA to actually look at age of individuals, using methylation patterns. We did look at the age of some of our bats, but we didn't really have GPS data corresponding with enough of them to really make too many inferences. And we also found that this bat is not a particularly long-lived bat. So we don't really have that much variation to maybe explain a lot of differences in behavior. But that is definitely a question that people should be asking in the future. I'd love to explore is how does age influence where they're going? And so the technology is there and we're starting to get into it.
I don't see any more questions. And maybe everybody could clap their hands together and thank Edward for his presentation.
Thank you.
The committee needs to reconvene. I would actually recommend that we take a half-hour break. So that would mean reconvene at 12:45. Our time, Eastern Standard Time. Is that okay for you, Yossi? Okay. So that way Edward can relax a little bit. We can have a bite.
Yeah.
And
Catch
then
my breath.
the committee should log into the defense Zoom account that I sent out. And I think you all have accepted that invitation. So thank you all for joining. I think I saw 82 participants at one point. So that's, I think, more than we would normally get. So that's pretty nice.
Thanks so much. Hope you all enjoyed it.
Good job, Edward.
Good job.
Thanks a lot.